All right, well, good morning, everyone. We are gathering today, this morning, to celebrate Dr. James Zuberbuhler with a special lectureship honoring Dr. Zuberbuhler. In the fall of 2019, we were very saddened by his passing. And once a year, we celebrate his legacy with an invited Grand Round speaker. For some of you who may not remember or know of Dr. Zu, I would like to just spend a few minutes summarizing his history and his extensive contributions to our field and our program. So Dr. Zuberbuhler was a local. He was born in a farm in Beaver County and did his undergraduate degree at Allegheny College, his medical degree and internal medicine residency at the University of Pennsylvania. He did cardiology fellowship at the University of Michigan and served in the military for 2 years, tour of duty in the Air Force. And although his training was in adult cardiology, his passion and calling was for the understanding of congenital heart disease. This is in an interview that Posca said. He said, congenital heart defects, in children, usually hundreds of different anomalies and hundreds of combinations. It's like putting together pieces of a puzzle. Very interesting and challenging. As an adult cardiologist, you're dealing with blood pressure, coronary disease. Well, it is interesting. It didn't seem as nearly as interesting as congenital heart disease. And so it was a clear choice I made. I had no regrets. I had a great career dealing with kids. His medical legacy was tremendous. He joined Children's Hospital Pittsburgh in 1963 and was the chief of cardiology from 1967 to 1994. He was acting chair of pediatrics in 87 to 88. He was an assistant medical director at Children's Hospital Pittsburgh from 82 to 94 and children's medical director 94 to 96 and vice chairman of the Department of Pediatrics in 88 to 94. And a very active member of the board of Children's Hospital and member of the executive committee. Here are some pictures of him. And, his training in education, in mentorship were tremendous, his contributions in that area. He trained and mentored entire generations of medical students, pediatric residents, pediatricians, and cardiology fellows. And you can see here, surrounded by the team, years back in the center, Cora Lennox, whose heart collection under her name we house here with more than 3,000 specimens, incredible congenital heart defects to learn from. And also here you see a young Dr. Bierman, and we also see Dr. Park, who contributed for the blade. I don't know if you can point to that in that right lower corner for the blade septotomy, and Dr. Fisher, among others. He was really a master clinician with a passion for clinical diagnosis, an incredible leader, teacher, usual clarity, intelligence, and brevity. A nurse that worked with him, Dawn Thomas, in cardiology for 19 years, she said, he reminded me of an old country doctor because he was very down to earth and very comforting talking to families. He never put himself on a level above families and patients or staff members. And truly, his contributions, in terms of scholarly work, were tremendous, and there are many landmark papers that have his name. Senior Geography of Epstein's Anomaly, Postoperative Electrophysiological Studies in Tetralogy, and the list goes on. And many of these we refer to in current papers on a regular basis. So the other thing that he did have a passion for is for skiing and the mountains. So he also not only for the sports side of it, but also he actually contributed to prevalence of congenital heart disease anomalies at high altitude with scholarly work in that aspect. And he summited Mount Whitney in the Sierra Nevada mountains in California, backpacked the Grand Canyon, and climbed Mount Imai, which is a holy mountain in China. And he had many interests, including golf, skiing, and an unsuccessful search for a giant meteorite. So, the key thing is the culture that we have here in Pittsburgh, I think, was very much something he said. Is the environment of camaraderie and team spirit that we have currently. That is really what persists today and that helps us succeed as a program. It's really what he started, and that's his legacy. Dr. Zuberbuhler is a local, and you can see was a local, and you can see here in this book, Pittsburgh Locals. There's a section on him. There he's with his horse Nellie, and there's a picture with his wife Jan, and he also contributed with an amazing description of all the wildflowers in Pennsylvania that you can actually find in that website. With beautiful pictures, which is really, really incredible. Over 900 species that he described. We are honored to have here today some members of his family, and we have Laura West, who is his granddaughter, who is one of our faculty in the Department of Pediatrics in Allergy, and his mom, Anne, and Jay, her dad, and Anne is Dr. Zuberbuhler's daughter. So wonderful to have you here with us. So every year we honor Dr. Zuberbuhler since 1995 with a prominent speaker in our field. And you can see, we've had, figures such as Dr. Freedom, et cetera. And today we're especially honored to have with us Dr. Shelly Miyamoto. Shelly is tenure professor in pediatrics at the University of Colorado School of Medicine. She directs the cardiomyopathy program. She also holds a chair in pediatric heart disease, the Jack Cooper Millisore Chair. And she's co-director and co-founder of Pediatric Cardiovascular Research Laboratory at the University of Colorado School of Medicine. She has an extensive number of awards, including significant NIH funding, and over 100 publications and high impact contributions. Her clinical expertise is in the care of children suffering from various forms of cardiomyopathy and heart failure, and spends much of the time performing research aimed to improve the understanding of the unique mechanisms that are involved in heart failure in children. So Dr. Miyamoto is here with us today. And she'll be presenting her Grand Rounds lecture on Does the Shoe Fit? Adult guideline-directed medical therapies and the failing pediatric heart. Shelly.
Well, good morning, and thanks so much to Jackie for this invitation. It's a real privilege to be here and to present this lecture in honor of Dr. Zuberbuhler. I've learned a little bit about him over the past couple of days, and I really think he obviously made a huge impact here and I think across our field. So certainly an honor. So the title of my talk, as Jackie said, is Does the Shoe Fit? Adult Guideline-Directed Medical Therapy and the Failing Pediatric Heart. I don't have any disclosures. So the objectives of my talk today will be to review outcomes and existing evidence basis for the failing pediatric heart and our contemporary approach to the treatment of children who are suffering from heart failure. I'll describe some age-specific findings in the pediatric failing heart that come from both our laboratory and others around the country. And then I'm going to review a cell-based model system that we've been using. It's really difficult to model pediatric heart failure. We just don't have good animal models like exist for adult heart failure. And that's really challenged our ability to understand the mechanisms of the failing pediatric heart. So I'll show you a cell-based model that we're using. And then the last few slides, I just want to discuss some challenges and opportunities related to child health research in general, specifically thinking about where is NIH funding, where are we getting the money to perform this very important research looking at pediatric specific diseases. So from what I know of Dr. Zuberbuhler, I think he actually would really appreciate this quote, which says, if it were not for the great variability among individuals, medicine might as well be a science, not an art. And my understanding of Dr. Zuberbuhler and just hearing a little bit about him is he treated every individual patient individually and personalized their treatment. So he was probably doing personalized medicine way, way before we even had that term, which I really admire and respect. But this is how I think of pediatric cardiology. We really are dealing with such a wide range of diseases, and each child really presents their own challenges. When thinking about heart failure specifically, much of what we do in our field of pediatric heart failure is we take randomized controlled clinical trials that are done in adults with heart failure. We take those medicines that have such a good evidence basis in adults and we extrapolate that treatment to children. And the question, that I'll kind of continue to pose throughout this talk is whether or not that approach is working. Is it appropriate for us to take adult heart failure therapies and extrapolate those to children without really thinking about the unique aspects that may be happening in these failing pediatric hearts? So when thinking about the characteristics of heart failure patients, when you compare adults to children, there are really dramatic differences. So the most common cause of heart failure in adults, of course, is ischemic heart disease. Whereas in children, we're dealing with children who have various forms of congenital heart disease or cardiomyopathies, which are diseases of the heart muscle. The average age of the adult heart failure patient is over 75 years, whereas the average age of our children that we're taking care of is less than 2. That's a pretty stark difference. And then when you think about comorbid conditions, there are most adult patients with heart failure have 4 to 6 comorbid conditions. So we're talking about obesity, diabetes, high blood pressure, whereas in children with heart failure, really the heart disease that they're suffering from is the main problem they have, and they don't have these other comorbid conditions. And how those comorbid conditions factor into how these medical therapies affect these patients, I think, is an important consideration. So when thinking about the evidence basis that the adults have, when adult cardiologists have when they're treating patients with heart failure, it's very extensive. So in this meta-analysis, if you look at the number of randomized controlled clinical trials of adult heart failure medications from 2001 to 2016, there were 118 randomized controlled clinical trials in that 15-year period. Over 215,000 adults were randomized and studied in these trials with a median of about 1,000 patients per trial. So let that sink in for a minute. 1,000 patients with heart failure enrolled in these clinical trials. Those are gigantic clinical trials and really does establish a very solid evidence basis. If a treatment is proven to be effective in these sizes of clinical trials, that's pretty convincing. If you look at the types of treatments that are studied, there are invasive therapies like ventricular assist devices, there are drug therapies, and then of course there are non-invasive therapies like exercise and nutrition and all these things that have been investigated in adult heart failure. So the next table I'm going to show you is the sum total of pediatric randomized controlled clinical trials in heart failure, and there are 3. So this is from the beginning of time. This is really the only randomized controlled clinical trials in pediatric heart failure patients. All 3 of these drugs were chosen to be studied in a randomized fashion in children because they were proven to be so efficacious in adults with heart failure. So these 3 drugs really decreased mortality, decreased morbidity, decreased heart failure hospitalizations, improved quality of life in adults with heart failure. If you look at the number in this column here, the number of patients that were enrolled in these clinical trials, it ranged from 116 to 360. And remember, the median enrollment in adult clinical trials is about 1,000 patients. So we just don't have the numbers. Fortunately, pediatric heart failure is rare, but then that makes it difficult to study. The outcome of these trials was that none of these drugs were proven to improve mortality or morbidity in children with heart failure. So these were mostly neutral clinical trials. That middle drug there, ivabradine, that's a medication that is aimed at lowering heart rate, and it did do that. So we proved in that trial that ivabradine did what it's supposed to do. It lowers heart rate in children, but there was no ability for us to study whether or not lowering heart rate in children really improved outcomes. So none of these drugs really have an evidence basis for which we can use to treat children with these medications. Irrespective of that, these medications are very routinely used to treat children with heart failure, despite the fact that these studies were neutral. Again, and we'll kind of go into some more detail about these studies and why I think people continue to use these medical therapies despite the lack of evidence in children. So what is goal-directed medical therapy or guideline-directed medical therapy in adult heart failure? What is the target? The cornerstone of guideline-directed medical therapy in adult heart failure really is to reverse remodel the heart. So when we talk about reverse remodeling, I usually think of remodeling as a positive thing. You're going to remodel your kitchen. That sounds like a really fun thing to do, but remodeling of the heart is not a good thing. So if you look at the picture on the left, that's a normal heart. The heart on the right has been remodeled because of heart failure. And what that means is instead of having this bullet-shaped left ventricle, the left ventricle becomes spherical and round. The walls thin out. The squeeze is decreased. The relaxation of that ventricle is impaired. So that's a remodeled heart. When looking at it at a more microscopic level, we see that the cardiomyocytes in these hearts are big. they're hypertrophied, there's fibrosis, so scar tissue is laid down in the heart muscle. And most of the guideline-directed medical therapy that's listed at the top there for adults is aimed at reversing this process, aimed at reversing cardiac hypertrophy or cellular hypertrophy, I should say. It's aimed at reversing that fibrosis or preventing ongoing fibrosis of the heart. So keep those things in mind, because the question is whether or not these remodeling aspects are happening in the pediatric heart. So again, looking at how effective are these drugs in adult heart failure, and I think it's pretty easy over the next couple of slides to convince you that these medicines are working and that they're really effective. So on the y-axis here is reduction in mortality. And so you can see across the top, those are the guideline-directed medical therapy drugs that are used to treat adult heart failure, ACE inhibitors, beta blockers, all the way across there. And you can see there's a decrease in mortality from about 15% with ACE inhibitors all the way up to a 33% probably improvement in mortality with beta blockers. So these are really, really effective drugs across the board. Looking at this in a little bit different way, I like this study because it demonstrates that as treatments have advanced and we've identified more and more treatments for adult heart failure, outcomes in adults continue to improve. So on the x-axis here is time since the diagnosis in months, and then on the y-axis is survival after the diagnosis of heart failure. The black line is the early era of the 1970s to early 80s, and then the very top line there, that purple line, that represents the most contemporary era of 2005 to 2016. And you can see there's significant improvements in survival as additional drugs and therapies have been developed over time. Really interesting and powerful stuff. I want to point out specifically what happens at about 5 years after transplant. I'm pointing this out specifically because I'm going to show you the equivalent data in children. But 5 years after the diagnosis of heart failure in adults, there's greater than 90% survival. So more than 90% of adults are alive and free from heart transplant 5 years after the diagnosis of heart failure. So keep that number in mind. So what about in children? Well, the latest iteration of the guidelines for the medical treatment of children with heart failure was published in 2014. That's now 9 years ago. Guidelines for adults are updated pretty much every 6 months to 12 months. They're updating the guidelines. We haven't updated ours in 9 years. There is an update that hopefully will be coming at some point in the relatively near future. But this is the latest set of guidelines. There were 23 recommendations for the treatment of heart failure in children with reduced ejection fraction. So that means 23 treatments for patients who don't have good heart squeeze. So patients with dilated cardiomyopathy, they have a reduced systolic function. 0 of those recommendations were based on level A evidence, meaning we don't have a single randomized controlled clinical trial to support the therapies that we're using to treat these children. 7 were level B evidence. And then 16 of the recommendations for the treatment of these children with heart failure were based on expert consensus. So you can imagine what the experts think. The experts really look at the adult randomized controlled clinical trial data and they say, okay, we think because these medicines work in adults, they're going to work in children. And that's really the basis for the heart failure treatment. And you know, admittedly, that's spelled out very clearly in these guidelines that we don't have an evidence basis. This is the expert opinion. And so that's really where our heart failure therapy comes from. So the question is whether or not that approach is working. Is this effective in the pediatric population? So I want you to focus just on the right-sided figure here. It's the one that says IDCM. That's idiopathic dilated cardiomyopathy. That's the most common form of cardiomyopathy we deal with in children. The left side is familial dilated cardiomyopathy. Those numbers are pretty small. But focusing just on the right-sided figure there, the purple and the green line are the time from diagnosis to transplant for children with cardiomyopathy. So again, the x-axis is time since diagnosis of heart failure. And then the y-axis is proportion of patients that suffer from an event. So the purple and the green line are time from transplant. The green line is the most recent era, and the purple line is earlier, the 1990s. So in these era differences, there's absolutely no difference. Okay, so we have not improved transplant-free survival in children with dilated cardiomyopathy. If you look at the blue and the red lines, those are time from diagnosis of heart failure to death. We have made some improvements there. So the red line is 2000 to 2009, and the blue line is 1990 to 1999. So across that era, there's been a little bit of improvement here in survival, or at least the lack of death in patients in children with heart failure. So a little bit of improvement. But when you look at what happens at 5 years after the diagnosis of heart failure, remember greater than 90% of adults are alive, transplant-free at 5 years. When you combine the transplant and death rate at 5 years in children, about 50% of children are they've either passed away or they've had a heart transplant 5 years after their diagnosis. So that's a pretty stark difference. And that really hasn't changed dramatically over time. You see this small change with the death rate, but really in general, this 50% rate of death or transplant 5 years after the diagnosis of heart failure is a pretty sobering statistic. So really, we know that pediatric heart failure outcomes are lagging behind that of adults. And there's now a growing body of evidence that perhaps we shouldn't be so surprised by this, that maybe there are differences in the failing pediatric heart that would suggest that we shouldn't expect them necessarily to respond in the same way to adult heart failure therapies. And I want to say a word of caution here. I don't want to imply that no children respond to these therapies. I do think there are children that respond to guideline-directed medical therapy, but I think we just have to be careful in expecting that it's going to be universal and that all children are going to respond. I think the proportion is probably smaller than what responds from an adult perspective. So I want to step back a little bit here and talk about this Carvedilol study that was published in 2007. This has really been kind of the center of how my research career developed, and it was really kind of the start of this, of at least our lab looking into how is the pediatric heart different than the adult heart, and it all really stemmed from this Carvedilol trial. So Carvedilol is a beta blocker, and it blocks both the beta-1 and the beta-2 receptor in the heart. And I'm going to show you on the next slide how this works and what the mechanism of that is. But really, this medical therapy I showed you in a prior slide, it resulted in a 33% decrease in mortality in adults with heart failure. This is a really powerful drug, and probably one of the most powerful drugs that we have to treat heart failure in adults. So back in the early 2000s, GlaxoSmithKline, which was the company that made Carvedilol, funded a pediatric clinical trial. So this was the first randomized controlled clinical trial in children with heart failure, was Carvedilol. Children with heart failure were randomized to either receive placebo or Carvedilol. And you can see from the figure on the right there that there was no statistically significant improvement in any of the outcome measures that were looked at in this trial, including mortality, morbidity, heart failure hospitalizations, all-cause mortality. This drug did not have the same effect in this pediatric population as it did in the adult population. So the conclusion from the trial was that Carvedilol did not significantly improve clinical heart failure outcomes in children. But there were a lot of challenges with this study. There's two in particular that I'm going to mention. One was that there was a greater than expected improvement rate in the children with heart failure. So children with heart failure tended in this trial to get better, whether they were on placebo or Carvedilol. They tended to feel better. They tended to have fewer symptoms. So that challenged the results of the trial. The other reason this was a difficult trial was that we enrolled patients both with dilated cardiomyopathy and with really complex forms of congenital heart disease like hypoplastic left heart syndrome. So this is a really heterogeneous group of patients, okay? And so the fact that we enrolled these different groups of patients challenged the study, because when they did a sub-analysis looking at how did the children with dilated cardiomyopathy do compared to how the children with single ventricle heart disease do, they found that children who had hypoplastic left heart syndrome and only one ventricle tended to actually do worse with Carvedilol than with placebo, whereas the children with dilated cardiomyopathy tended to do better with Carvedilol, but not statistically significant. We didn't have the power to really look at that. So when you put those two groups together, of course, you get no difference. So one of the big lessons that was learned from this trial is that we have to be really careful in our study design when designing randomized controlled clinical trials in children with heart failure. We can't have these very different disease groups included. And so subsequent clinical trials to this, that ivabradine trial and the Entresto trial that I showed you earlier, excluded patients with complex forms of congenital heart disease like hypoplastic left heart syndrome. Those patients are now being left out of these trials because it's really felt like they do respond differently. So that's one of the lessons we learned. Despite that, we're still struggling to find differences in our randomized controlled clinical trials, even when just including the dilated cardiomyopathy group, but it's still, I think, an important lesson. So because that study demonstrated no improvement in children with Carvedilol therapy, it raised the question of whether or not there may be something going on in the hearts of these children that would make them not respond, that maybe this is a real biologic finding. There's something different about these failing hearts in children that would make us believe that this is a true finding, that Carvedilol really does not work. So one of the things we wanted to do was look at adrenergic receptor expression in the hearts of these children. So just to remind us of the biology of the adrenergic system, there are two main beta receptors in the heart, the beta 1 and the beta 2 adrenergic receptor. They're pictured here. In the setting of heart failure, there's increased catecholamines, adrenaline, norepinephrine. That is part of the heart failure phenotype, and that means that these receptors get stimulated. And initially, that's a compensatory response, so sort of a fight or flight response. You have an increase in adrenaline, you mediate this pathway where you have an increase in cyclic AMP expression that activates protein kinase A, which then phosphorylates calcium proteins such as phospholamban, and you get increased contractility and increased relaxation of the heart muscle. So this is a compensatory response and is a good thing. But over time with heart failure and chronic stimulation of those beta receptors, we know that in adults with heart failure, you start getting desensitization and internalization of that beta 1 receptor. So this receptor is just getting hammered with these catecholamines, and eventually the receptor stops responding to those and it starts internalizing, and the expression is decreased. And it also starts mediating a different pathway, this more detrimental pathway mediated through CAM kinase, which results in some of that remodeling of the heart we talked about, where the heart gets big and doesn't squeeze well. There's dephosphorylation of the calcium regulatory proteins like phospholamban, so you're not getting that response of improved contractility. Instead, you're getting diminished heart contractility and decreased cardiac output. So this beta receptor is now mediating this very pathologic pathway. There's apoptosis involved. It's really a detrimental response. The beta 2 adrenergic receptor up top is really a very different receptor. It's more of it mediates a more positive response, and it's a pro-survival anti-apoptotic mediated pathway for the most part. And that receptor doesn't really change in adult heart failure. The expression of that receptor stays fairly stable, and I'll show you some of that data. So that's where Carvedilol comes in. So the goal of Carvedilol is it's going to block both the beta 1 and the beta 2 adrenergic receptor, and it's going to prevent this pathologic pathway that's being mediated through the beta 1 receptor. So that's really why Carvedilol is effective. So back in 2017, once this Carvedilol trial was published, I began to collaborate with two colleagues in adult medicine, Brian Stauffer and Kika Sukharov. And Brian and Kika had worked in the lab of Mike Bristow at the University of Colorado. And Dr. Bristow really was one of the pioneers of the use of beta blocker therapy in adults with heart failure. And so when these two adult cardiologists heard me telling them that Carvedilol was not effective in children, they actually just didn't believe it. They're like, this just can't be the case, right? This is a miracle drug. It should work. We don't understand why it worked. And so the three of us started working together to really try and better understand the beta receptor system in the failing pediatric hearts. And so our hypothesis was that maybe there are age-related differences in how this beta receptor responds in children and adults, and that could potentially contribute to the clinical differences we saw in that Carvedilol trial. So to investigate this, we started by using our tissue bank. So at the University of Colorado, we have a really large tissue bank that contains hearts from both children and adults with heart failure. So every child and adult that gets listed for a heart transplant is consented by our team. And if the family and the patient agree, we're able to take pieces of the heart at the time of heart transplant. So the heart is removed by the surgeons, the diseased heart is removed by the surgeons. We can collect parts of that heart and store that in our biobank for future studies. So what we did in this study was we took pediatric hearts from children that were less than the age of 12, and we chose that age because we were trying to avoid puberty. All sorts of crazy things happen in puberty, as everyone in this audience knows. So we wanted to avoid that time frame. So we had children with idiopathic dilated cardiomyopathy, and then we took hearts from adults with idiopathic dilated cardiomyopathy, really the most apples to apples comparison we could do. We excluded patients with congenital heart disease, we excluded adults with ischemic heart disease, and really just looked at idiopathic dilated cardiomyopathy. We had age-matched non-failing controls. Where these control hearts come from, these are non-failing normal hearts, and they come from pediatric and adult donors whose heart could not be placed with the recipient because of size or blood type mismatch. Really, really rare for us to have pediatric hearts that don't get placed, but it does happen every few years. We'll get called by Donor Alliance to say we've got a pediatric donor. The heart function is great. We have no appropriate recipient within, our geographic area. And so we're able to collect that heart tissue as our non-failing control. So that's where those control hearts come from. So when you look at these figures here, the pediatric heart expression of these beta receptors is on the left and the adult is on the right. The clear column is non-failing control, and then the black is the idiopathic dilated cardiomyopathy expression. So that first graph is total beta receptor expression. And what you can see is that both in the pediatric and the adult hearts, there's decreased total beta receptor expression. So really no difference there. You go to the next, that middle graph there, that's the beta-1 adrenergic receptor. I don't know if my pointer works here. So this is the beta-1 adrenergic receptor. And as I told you, we just kind of recapitulated what had previously been shown in adult heart failure, that the beta-1 receptor is decreased in adult heart failure. And we saw the same thing in the pediatric hearts. Okay, so far, no difference. The differences come when you get to that beta-2 receptor. And remember, that's a good receptor. It's pro-survival, anti-apoptotic. There's no change in the expression of the beta-2 adrenergic receptor in adults, but there was a decrease in this beta-2 adrenergic receptor in the children. And that's a very different response than we saw in the adult population. So what does that mean? What does it mean? Why is this important that the beta-2 adrenergic receptor is downregulated in children? Well, this is a good receptor in general, and our hypothesis was that maybe with Carvedilol, which blocks both the beta-1 and the beta-2 receptor, we're blocking that good beta-2 receptor below a threshold that may be important. Okay, so it's already downregulated. Now we're going to block it further with Carvedilol. Is that causing a detrimental response? So there are many different kinds of beta blockers. There's Carvedilol, which blocks both the beta-1 and the beta-2 receptor, but there's also beta blockers that only block the beta-1 receptor. Those have been shown to be efficacious in adults with heart failure as well. So our hypothesis was maybe if we don't block the beta-2 receptor in children, that would be better. Okay, let's just keep that beta-2 receptor okay, and let's only block the beta-1 receptor. So our hypothesis was that maybe beta-1 blockers would work in children and adults, but that non-selective blocker like Carvedilol would only work in adult populations. So how do we study that? Well, we developed an animal model. So we have a young mouse and an old mouse. And we put isoproterenol infusion pumps in these mice. So this isoproterenol is basically a catecholamine. Okay, so we're going to recapitulate this kind of catecholamine-induced heart failure. We're going to do that in the old mice and the young mice, and then we're going to give them different beta blockers, and we're going to see what the response to those beta blockers is. The outcome that we used was heart rate, body weight. So what happens in these mice when they get stimulated with that catecholamine is that their heart rate increases. Okay, that's the remodeling that we see, that hypertrophy of the cardiomyocytes and growth of the heart muscle. So when you look at this figure here, there's old mice on the left, young mice on the right, placebo. So these are infusion pumps that aren't producing anything other than just saline. But then when you put the isoproterenol infusion pump in, you can see the heart rate, body weight increases. And that happens in both the young and the old mice. When you give the mice Carvedilol, you can see that in the old mice, you get a decrease in heart rate, body weight. So really effective. When you look at what happens in the young mice, just like we saw in the clinical trial, there was no change in heart rate, body weight with Carvedilol. So what happens if we just use a beta-1 selective blocker? That's metoprolol, only blocks the beta-1 receptor, leaves that beta-2 alone. And you can see that there was an improvement in heart rate, body weight, both in the old and in the young mice. And really interestingly, you can see that Carvedilol was better in the older mice than metoprolol as far as decreasing heart rate, body weight. It brought it all the way back down to the baseline, whereas in the metoprolol mice, just like in clinical trials, metoprolol is not quite as good for adults with heart failure. It's not quite as good as old mice, apparently, with heart failure either. But in the children, it really did work. It didn't bring it down to into the normal level, but in these young mice, it actually did blunt that heart rate, body weight response. So, people have asked me over the years, well, does this mean we should be treating children with just metoprolol? I would not say that. These are mice. I don't know how this would translate to humans, but I just, I think from my perspective, this is just thought-provoking more than anything. It's it just makes us, I think for me, it just makes us continue to think we need to be really critical in how we think about these patients and not just make assumptions that everything's going to be the same in an adult and a pediatric patient population. So what about other aspects of remodeling? We talked about the beta receptor system. One of the other really important targets of heart failure therapy in adults is fibrosis, and that scar tissue that's laid down in the hearts of adults with heart failure. So Casey Wolf, pictured here, was a postdoc in our lab and now is a junior faculty in adult cardiology, and she did a study a few years ago where she took heart tissue from our heart tissue bank and she stained this tissue looking for evidence of fibrosis in the pediatric hearts. And what she found was that there was a lot less interstitial fibrosis in the pediatric failing heart compared to the adult heart. This data was really interestingly, we didn't even know this was happening, but one of my now collaborators, I didn't know him at the time, but Corey Levine from Washington University in St. Louis was doing a very similar project with a fellow of his, Meghna Patel. They were doing a very similar study, and we didn't even know it. Both of these studies got published in 2017, and they saw the same thing. So if you look at Corey's study over here, they've got pediatric donor hearts, adult donor hearts, and then you can see in the dilated cardiomyopathy hearts, there's much less fibrosis in the pediatric hearts than in the adult hearts. And we did this really elegant design where they actually quantified the fibrosis. And we demonstrated that as you increase in age here with their dilated cardiomyopathy hearts, you do get increasing fibrosis. There is just less fibrosis in the younger hearts. One of the questions that we answered, and I don't show the figure here, but one of the questions we answered is, does this just have to do with amount of time that you have heart failure? So if you have heart failure for a longer period of time, are you more likely to develop fibrosis? And at least in our tissue bank, that did not seem to be the case. So even if you take an adult who was transplanted 2 years after the development of heart failure and a child who was transplanted 2 years after the development of heart failure, the amount of fibrosis in that adult heart was higher. So we just don't know if targeting fibrosis in the pediatric heart really is as efficacious as it is in adults. Again, that being said, there are people in this room, myself included, who have definitely seen significant fibrosis in pediatric heart failure. So this is not a universal finding, but I think there are children who don't have as much fibrosis, so may not respond to medications targeting the renin-angiotensin-aldosterone system, such as mineralocorticoid receptor antagonists or even drugs like Entresto. So the other thing I showed you was we talked a little bit about hypertrophy of cardiomyocytes. So what happens with the remodeling of cardiomyocytes in children compared to adults? We know that cardiomyocyte hypertrophy is a hallmark of cardiac remodeling in adults with heart failure, but this also differs by age. So again, we were doing a separate study looking at this at the same time. Corey Levine and his group was doing this. We're doing this in WashU, and again, we found the same thing when looking at our tissue banks. So this figure down here on the left is from Corey's lab. So pediatric donor, adult donor, and then when you look at the pediatric cells, they don't hypertrophy to the same extent that the adult cells do. And we found the same thing up here. So here's adult and pediatric cells, and you can see that the adult cells are really big and globular looking, and then the pediatric cells really don't look any different than donors. So again, we're not seeing that cardiomyocyte hypertrophy to the same extent in the pediatric hearts as we are in the adult hearts. So another important difference. So what about, are there drugs that may work in children that don't necessarily work in adults? So kind of flipping this paradigm on its head. And I think there are. And one of the examples of a drug that a lot of us in this room have experience with is milrinone, which was a phosphodiesterase 3 inhibitor. Milrinone was studied many years ago. So back in the early 1990s, there was a study published in the New England Journal of Medicine called the PROMISE trial. So this was long-term milrinone or chronic milrinone therapy in adults with heart failure. And this study demonstrated there was actually a 34% relative increase in cardiovascular mortality in adults with heart failure. Around this time, there was also the development of a drug called enoxaparin, which was an oral phosphodiesterase 3 inhibitor, but that drug ended up going away because of the PROMISE trial. So in other words, we don't have access to any oral PDE3 inhibitors because these drugs were deemed to be ineffective and actually dangerous in adults with heart failure. So unfortunately, this drug is not available any longer in this country. So what about in children? Well, a lot of us in this room have used milrinone pretty chronically for many months in children with heart failure. It improves their symptoms. This has been written up in small case series. We do not have a randomized controlled clinical trial, so we have to be a little careful about how we interpret this drug. But a lot of us have used this drug, and I've sent patients home on this drug. It's an infusion, so they have to have a long IV in, and they need to have this drug given to them 24 hours a day through an infusion pump. They've got to carry around a little fanny pack with a pump. It's really inconvenient. There's no oral therapy. So this is not something we can use long-term in children. We can't have kids walking around with a PICC line and an infusion pump forever. So it's really just used as a bridge to transplant or used as a therapy to get a child out of acute decompensated heart failure. But it has been successfully and safely used in children. So again, looking back at our tissue bank, we wanted to answer the question of whether or not there may be a biologic reason that this is the case, that this drug is not effective in adults and actually dangerous in adults and efficacious in children. So back to the beta receptor system, you've got increased catecholamine stimulating the beta 1 and the beta 2 adrenergic receptor. We're going to look at this different arm of the pathway. So remember, we talked about this good arm of the pathway where you get increased cyclic AMP, increased PKA, activity which phosphorylates this very important calcium regulatory protein called phospholamban. When phospholamban is phosphorylated, you get improved uptake of calcium and improved contractility of the heart. PDE3, which you can see here, is an enzyme that breaks down cyclic AMP into 5' AMP. So when you break down cyclic AMP, now you no longer are getting this PKA-mediated pathway. So the idea behind phosphodiesterase 3 inhibitor therapy is that if you can prevent PDE3 from breaking down cyclic AMP, you can actually restore some of the cyclic AMP and continue going down this good pathway. So that's how phosphodiesterase 3 inhibitors are supposed to work. We want to see an increase in cyclic AMP, an increase in PKA activity, and increased phosphorylation of phospholamban. Those would all be good things. So what actually happens in the hearts of children and adults? This was a study done by, she was a fellow at the time in our lab, Stephanie Nakano. She's now a mid-career faculty in our department in Colorado, so still doing really good work. But she did this study as a fellow where she looked at cyclic AMP expression in children and adults with heart failure. So again, back to our tissue bank, looking at idiopathic dilated cardiomyopathy specifically. And you can see that both in the pediatric, which is on the left, and the adult on the right, there's a decrease in cyclic AMP in the setting of heart failure. Okay, so when you take these hearts and you look at cyclic AMP expression, it is definitely diminished in the failing heart. So what happens if you take the heart of a child who's been treated with milrinone before transplant? So that's this group. So these are failing patients treated with milrinone before they were transplanted. And you can see there's partial restoration of cyclic AMP. So this was a statistically significant improvement in cyclic AMP expression over the failing hearts in children. Not all the way back to normal, but an improvement. When you look at what happens in the adults, there was no change. So they do not get this augmentation of cyclic AMP as we would have expected. So what about downstream effects of cyclic AMP? What about phospholamban phosphorylation? So phospholamban phosphorylation is decreased in the failing hearts of children and adults. In the treated group, we saw recovery of this phospholamban phosphorylation in the pediatric hearts, but not in the adult hearts. And so, again, just a little bit of caution here. This does not necessarily prove anything, but there's an association here that suggests that milrinone may have a different effect in the pediatric hearts than it does in the adult hearts. And could this be part of the reason why milrinone is so effective in children and not effective in the adult population? So we'd love to get enoxamine, which is the oral PDE3 inhibitor, back so that we can use that to help treat children. And maybe we could do a randomized controlled clinical trial of that medication in children, but that's challenging. So I had this slide in here before, but I didn't realize that Dr. Zuberbuhler was a wildflower enthusiast, so he would have really appreciated this picture. But the purpose of this picture was really just to kind of help us realize that we're kind of looking at characterizing these pediatric hearts at a larger level, and what happens when we try and dig down deeper and really start looking at kind of gene expression and other factors in these hearts that could be explaining why the pediatric heart differs so much from the adult heart. And so we have done a couple of studies that I think will help generate some hypotheses about the mechanisms of pediatric heart failure and what makes it so unique. And I'm going to show you one of those studies here, which is an RNA sequencing project that we did in adult and pediatric heart failure. So in this study, what we did is we took adult dilated cardiomyopathy hearts and adult control hearts, and we performed this very comprehensive gene expression panel in them, and we looked at what genes are different in the adult failing heart, and then we did the same in the pediatric heart. What genes are different in the pediatric failing heart compared to the non-failing pediatric heart? And then we compared these two groups together. And what you can see here is that when you look at the genes that are upregulated in the failing hearts, there are only 48 genes that are similar between the children and the adults. Looking at downregulated genes, there's only 56 genes that are similar between the failing adult and pediatric hearts. Lots of genes are different between these groups. So this really suggests that the pediatric failing heart is a distinct disease process. These are just really different diseases. When we look at what pathways are associated with these genes that are differentially expressed in the pediatric heart, there are things such as dedifferentiation, which is something that I think is really interesting and actually something that I think is worth exploring further. There's increased fetal gene program expression, pathways related to decreased heart function, decreased hypertrophy, decreased apoptosis. Those are things that are predicted, which is really different than the pathways that are involved in the failing pediatric heart. So again, this is really just mainly hypothesis generating and helps us kind of try and identify what pathways we might want to be thinking about when it comes to understanding mechanisms of pediatric heart failure and potentially identifying unique therapeutic targets. So how do we get at this further now that we have, we understand what some of the genes and pathways are in these pediatric failing hearts? How can we get at this? Get at what mechanisms are involved, what model systems can we use? And this is really, really challenging. We can't perform the invasive studies that have been so beneficial in adult heart failure. So for example, Dr. Bristow, when he first started thinking about beta blocker therapy, did this really cool study where he biopsied the hearts of adults, and he looked at this gene expression pattern that shows this very pathologic gene expression pattern, treated those adults with a beta blocker, brought them back to the cath lab after 6 months, biopsied them again, and showed that the gene expression pattern had changed and was now more normal. That's a really cool and very elegant study. We cannot do that in the pediatric population. I think that would be very difficult to get by an IRB to be doing biopsies in the hearts of children. So we just need different model systems. We know the randomized controlled clinical trials in children are challenging because of the rarity of the disease. And we know that that animal models are really difficult. We've got the mouse model we developed. It's certainly not perfect. And so we really need to figure out how can we study both cardiomyopathies and congenital heart defects in children in order to kind of get at some of these mechanisms. So I'm going to tell you a story about a model system that we're currently using and kind of how we got there. It's a really cool and interesting story. So we got inspiration for the model that we're currently using from Dr. Leslie Leinwand. And Dr. Leinwand is a molecular biologist at the University of Colorado in Boulder. And she's got this longstanding interest in understanding physiologic versus pathologic hypertrophy. So physiologic hypertrophy is what you get in hearts of athletes, for example. So she's looked at exercise and what is it about exercise that mediates this very healthy physiologic hypertrophy. And then she's looked at sarcomere gene variants, for example, as examples of pathologic hypertrophy. But she's got this interest in hypertrophy of the heart. Well, many years ago now, she was on vacation in Burma. And the tour guide that she was with was telling her that python hearts increase 40% in size about 48 to 72 hours after they eat. So these animals only eat every several weeks, but when they eat, they have this marked increase in heart size, and then the heart shrinks back down to normal as they fast, they eat, and the heart increases again. So she was completely fascinated by this. And so fascinated by this, because this is clearly physiologic hypertrophy, this is healthy and normal, she decided to develop a python lab. So no kidding, she developed a python lab up at CU Boulder, brought all these snakes in and started studying this physiologic hypertrophy. Her postdoc at the time, Cecile, she had many postdocs, always has many postdocs, but Cecile Requelme was the one postdoc that was brave enough to study these pythons. So Leslie and Cecile studied these python hearts. And this is what the hearts look like. So before the meal and then 3 days after the meal, you get this 40% increase in cell size, in heart size. So in an effort to try and understand what it was about these animals that was causing this increase in size, they drew blood from the pythons after they had been fed, and they took the serum from these pythons that had been fed, and they used that serum to treat neonatal rat ventricular myocytes, so primary myocytes. They put the serum on the myocytes, and they saw that these myocytes increased in size in a very physiologic manner. They also took serum from the fed pythons and they gave it to the fasting pythons. So they injected serum from the fed pythons into the fasting pythons, and the fasting python's heart grew. So there was something in the circulation of these animals that was causing this physiologic growth. And ultimately, to make a long story short, and you can read the paper in Science back in 2011, they found that there were fatty acids in the serum that were causing this physiologic growth. So it is a really cool story. Well, my colleague, Kika Sukharov, had done a postdoc in Leslie's lab, so she was following all of Leslie's work, and she had the idea that, why don't we try this with pediatric heart failure serum? So let's take serum from children with heart failure and let's put them on neonatal rat ventricular myocytes and see if there's something in the circulation of these children that might be causing, that might be potentiating the cardiovascular effects. So our hypothesis was very similar to what Leslie found, is that circulating factors contribute to the heart failure pathophysiology, and that serum-treated cardiomyocytes might serve as a novel model to study mechanisms of disease without having to biopsy pediatric hearts and just drawing blood, which we can do in the pediatric population. So we looked at pathologic gene expression in these hearts that were in these cardiomyocytes that were treated with the heart failure serum. So BNP and ANF are pathologic genes that were both increased in the cardiomyocytes that were treated with serum that came from children with heart failure. This is compared to non-failing normal control serum. We've looked at many different things, or tried to look at many different things within the circulation. We haven't been as lucky as Leslie in finding a specific fatty acid or anything that we can identify specifically that's causing this pathologic response, but we continue to look. And one of the things that we've been interested in is exosomes, and whether or not there are exosomes in the circulation of these children that might be potentiating this pathologic response. So exosomes are these microvesicles. They're 40 to 100 nanometers in size, and they package all sorts of things. These are really kind of cool little microvesicles. They package proteins, microRNAs, mRNAs, all sorts of things are in these exosomes, and they can carry that packaged material from one cell to another. And so we wondered if maybe there was something in the exosomes of these pediatric patients that was resulting in this pathologic response in the myocytes. So we're able to do a precipitation process where we can isolate these exosomes, and then we could use this nano-tracking software to identify these exosomes, and we could count them and make sure that we are indeed isolating exosomes. We can look at their size. And what we found was that in the patients with dilated cardiomyopathy, they actually had fewer exosomes than patients with normal children. So it's not the number of exosomes that's causing this pathologic response for sure, because children with dilated cardiomyopathy have fewer exosomes, but it may be something within those exosomes. So the next thing we did is we labeled the exosomes with a fluorescent antibody so that we could track them. And we put the exosomes on these cardiomyocytes and we watched for uptake. And you can see over 24 hours, you start seeing the exosomes coming into the cardiomyocytes. We proved that these exosomes were not just binding to the cell membrane of these cardiomyocytes, they're actually inside the cardiomyocytes because we washed these cells. And when you wash the cells, you still see them light up. If they're just sitting on the membrane, they would have gone away. So these exosomes are clearly getting inside of these cardiomyocytes. So what happens when you block the exosomes from getting into the cardiomyocytes? Can you prevent that pathologic response? And you can. And we did that by treating the cells with something called Site D, which prevents exosome uptake. So if you look at these figures here, the ANF and the BNP are increased. This line is non-failing control expression of these genes. When you treat cardiomyocytes with exosomes from children with heart failure, you get an increase in ANF and BNP. If you block the uptake of those exosomes with Site D, you don't get that pathologic response. So this suggests that exosomes could at least be contributing to this pathologic response in these cardiomyocytes. So I just want to finish up here in the last few minutes. Where do we go from here? We know now that age and etiology of heart failure are important factors in the identification of appropriate therapeutic targets. I think that all of us in this room probably agree that children deserve focused study, and that perhaps in the case of heart failure, we shouldn't just entirely rely on guideline-directed medical therapy that is founded in adult populations. That yes, that's probably effective in some patients. I'm not saying that we shouldn't be using it, but do we need to think broader? Do we need to think about identifying more novel therapeutic targets based on what we're seeing in these failing pediatric hearts? But this is challenging, and I just wanted to finish with the last few slides kind of talking about how do we get the money to do this? Where is this funding coming from to study pediatric heart disease? It's rare. Pediatric heart disease is rare. And when you think about how much funding from the NIH goes towards the study of pediatric diseases in general, this is not specific to cardiology or pediatric heart disease. This line here represents the percent of the NIH budget that's allocated to pediatric research, and it's only around 12%, and that really hasn't changed over time. So not much funding from the NIH is really going towards pediatric research compared to adult disease. We look at the percent of contribution of each institute within the NIH towards pediatric funding. NICHD, which is really the premier pediatric focused research institute, only provides about 18.5% of funding towards pediatric research. There's all these other institutes that are also contributing. The pay line is very challenging. Obviously, these are incredibly competitive awards to get. The K award priority score for an NHLBI-K right now is 28, and only the R01 pay line is 15%. So it's really, really difficult to get these grants. And in the pediatric realm, of course, it's a challenge. There's also been quite a bit of contraction of where this funding is going. So 63% of R01s are granted to 15 institutions. You can see I highlighted University of Colorado here with 61 of the pediatric R01 equivalent awards when this article was published in 2018. But University of Pittsburgh is right here at 57, so we're kind of in that same realm. And when you look at cardiology specifically, at least at the time that this article was published, there were only 36 pediatric R01 awardees in the field of cardiology. That's less than one per state. It's just, it's a really challenging thing. And so I think, my point here is we just need to continue to encourage our physician investigators and encourage our PhD investigators that are interested in child health research to continue pushing those boundaries, continue advocating for increased funding for pediatric research, because it is difficult and we just don't have a lot of people doing it right now. So I think for the sake of time, I just want to wrap up here and really just hopefully bring home this point that I do think children deserve focused study, and that's not specific to just heart failure, that I think there are lots of pediatric diseases that we really have to be thoughtful about and make sure we're investigating those specifically. So thank you very much for your time, and I'd be happy to answer a couple questions. If anybody's ever in Colorado, please don't hesitate to let me know. I'd be happy to give you a tour of our campus and of our children's hospital. Thanks very much.